All right. Good evening, everyone, and welcome to Thursday night here on MCAT Free Prep Week. We are super excited to have you with us tonight. I'm super excited, and I know that our team is super excited. We were just chatting before we got started, and we know you guys have done a lot of work this week. For those of you that have been attending multiple sessions, I saw from the poll or from the chat, I should say. That for a couple of you, this is actually your first time here this week. So either way, we're super excited to have you joining us. And tonight, we're going to do things a little bit differently. I know for those of you that have been here before, we've had, kind of a guest teacher come on and all that good stuff. I'm going to actually be with you for the whole 2 hours. So super excited about that. And super excited for our team who's here as well today. Adriana, Ashwarya, Deborah, Carrie, and Nikolai. So these are all folks that you're going to be interacting with kind of throughout the course of the next 2 hours. My goal here is for you to learn something, not only from the scientific perspective, but also to learn about yourself as far as the next steps that you need to be taking for your MCAT preparation. This is a huge exam, and there's no doubt about it that there's an importance that needs to be placed and kind of a gravity as far as how you are preparing for this exam. And that's what we're here to do tonight, is to talk about that. We're going to do some molecular genetics. So we've got some DNA and transcription for you kind of coming up. So lots and lots of fun as we go forward. My name is Petros Manassi. I'm the director of pre-health programs here at Kaplan, but I actually wear two hats. And my second hat is that I am a member of our MCAT faculty team, along with Ashwarya and Adriana and Nikolai and Deborah, who are here with us tonight. And I forgot somebody, Carrie. Carrie, how could I forget you, Carrie? I'm a proud member of that team, and get to continue to teach students on the MCAT channel. Have been doing so for the last 18 years. So I've, we were talking about this. I think between the six of us, we've got over 40 years or so of MCAT experience among us. And so I want you to really tap into that tonight as far as carving out your path and carving out your journey. I was just actually at the AMC conference. Those are the folks, of course, that write the MCAT and administer the MCAT, and saw, some interesting stuff in terms of statistics and things that, publicly available, but it's kind of cool to see them from the folks themselves. And so if you got questions about admissions and all that good stuff, more than happy to guide you along the way and to answer those questions kind of as we go. So as stated on the slide here, tonight is inside the channel, looking at the MCAT and looking at some material. So real quick, as far as our agenda, we're gonna do a little bit of an overview of the MCAT itself, get your questions answered. So if you've got questions, we've got answers. And so I want you to be typing those into the chat as we go. We're going to do an MCAT channel session. So as I said, tonight is all about DNA and transcription. So good old molecular genetics. So we'll spend about an hour or so, maybe a little bit more, just depending on our pace and our time, talking about that and going through literally the same session that we teach on the MCAT channel. The MCAT channel is something that is included as part of our MCAT programs. We have over 90 or so sessions and adding more as we speak to the MCAT channel. Those are live instruction sessions that are taught by literally our top 20 or so faculty at all of Kaplan. And then at the end, we're going to give you an opportunity to just get your questions answered. So, I was talking with the team was we've got a lot of experts here. And so really tonight's going to be about you getting all your questions answered. I'm actually going to show you a little technique that we do in our sessions and with our students, which is the idea of how to categorize your wrong answer choices and your questions that you've gotten wrong, and how you can use that to learn and identify patterns to help improve your score. So we'll talk about that towards the end as well. A couple other fun things for you to all know, and I wanted to make sure that you knew this before we dive in, is that number one, as a participant in MCAT Free Prep Week, I'm going to give you guys a code, and I want you to write this code down. It's going to be good for a little bit more time. It's good until the end of the week, so basically until the weekend. And that is, the code is MCAT 250. that is a $250 savings on our MCAT programs that you can use. And that is special. That is one of the biggest savings that we have for this year. And that is special specifically for you, individuals who have taken the time and dedicated the time to come into come to MCAT Free Prep Week. And then the other thing, which we're really kind of excited and happy to announce, and something that we've been putting together for some time, and we wanted to launch it during Free Prep Week, is that we've actually launched a free trial of our MCAT program. So you know you can sign up for Netflix or Hulu or whatever, you get a little bit of a free trial. We have that now for MCAT programs because I firmly believe that the best way to see how our MCAT programs work, I can talk about them all day, is to actually get a flavor and a taste of it yourself. So the link is posted at the top of the chat. You can go there. It's a 7-day trial. It gives you a sampling of all the different resources. The real cool thing about it is that it's actually designed to be just about the amount of work that you would need to complete in a 7-day period. So I encourage you to use that. You can hang on to that link, sign up for it later if you don't want to sign up for it necessarily right now. But I'd encourage you to do it sooner than later because there's a diagnostic exam that is built into the free trial. So you can actually see where your score would be. And then once you get enrolled into a course, we'll actually port over those diagnostic results and actually start to populate your recommendations for your MCAT preparation. The recommendations actually tell you, here's what you're good at, here's what you're weak at, but more importantly, here's what you should start working on right now. It actually points you to specific resources built into our program. So highly encourage you to take a look at those as the days kind of start to go forward. So that's something that's important for you, and definitely to kind of keep in mind as you're looking at it. So with that said, the free trial, yeah. So I saw a couple questions come in the chat. I'll just address this last thing before we dive in. The free trial is not just sessions. It's actual, it's the lessons from our course. There are chapters from the books. There are practice questions, practice quizzes, the science review videos, which are these short little videos. So it's literally a sampling of our overall program. When you think about your MCAT preparation, there's the class time, and then there's all the other stuff that you have to do. And all the other stuff is actually more than your class time. You're spending on average 300 to 350 total hours preparing for your exam. A lot of that is spent doing practice. Doing well in the MCAT is about learning science, but then knowing how to apply that science. In fact, the MCAT doesn't reward you on just knowing content. It rewards you on your ability to apply that content. So really, really important to kind of keep that in mind. A couple of you are asking if the free trial includes full-length exams. It does not. It includes the diagnostic exam, which is half exam. The full-length exams are included as part of the course or part of our prep packs if you were interested in getting one of those. But the free trial is just that. It's a free trial of what our programs look like to give you a taste and get you kind of looped into what you're going to be experiencing, and it's completely free. So I'd encourage you to sign up for it. It's not a bad thing to have for at least for 7 days time as you're looking at it. So with that said, let's take a look at the MCAT. I know a lot of you have seen this before, but I wanted to take a little bit of a different approach on this for you, and that is, when you look at this exam, it's a long exam, and I can probably guess that not many of you, unless you have taken the actual MCAT, normally on a regular daily basis, sit down for 7-hour exams. I don't think I would be wrong in making that assumption, right? That's not something that we normally do. It's not kind of part of our normal rhythm of life. Final exams in school are maybe 3 hours, sometimes 4 hours if they're, particularly harder instructor or particularly harder course. But the reality is that we don't spend 7 hours sitting for exams. And the other thing that we don't do, which the MCAT requires us to do, is actually sit for an exam that has such a wide variety of subjects. In fact, on the MCAT, you are tested on a total of 8 subjects throughout the course of the day. And in each of the 3 science-based sections, you are tested on multiple subjects, more than 2 subjects at a time. In the chemical and physical foundations of biological systems, you're tested on chemistry, you're tested on physics, you're tested on OChem, on biochem, and on biology on top of all of that. So the test is not just about, the length of the test. It's not just about the questions. It's also a lot about the integration and the application of the sciences. And I'll give you a very, very simple example, a very common type of question, a very common example, I should say, of the integration of the sciences is when you think about the circulatory system. The circulatory system is really nothing more, when you think about it at the very basic fundamental scientific level, it's nothing more than a pump that is pumping a liquid through a series of pipes. Those pipes happen to be arteries and capillaries and veins, and the pump happens to be the heart, which is a very complex organ that we have. And of course, our circulatory system, the liquid is nothing to be frowned upon. It's blood, which has got all sorts of kind of components in it. But really, at the very core fundamental basis, it is a pumping system. And the MCAT loves to test you on that. It loves to test you on something like the circulatory system, but in the context of the physical sciences. So it is not unheard of to see a question that relates to the circulatory system that is actually asking you to apply physics formulas. And these are formulas that you maybe aren't used to applying to a biological setting, but something that is very, very much reasonable and very okay for the MCAT to be asking you. So I don't want anybody to kind of be fooled into thinking that, oh, I just got to study the sciences. In fact, one of the worst things that you can do is only use the books that you have to review your material, because your books from your college classes, because really that's not what the MCAT at the end of the day is going to reward you on. They're not going to reward you on just knowing information. They're going to reward you on the ability to actually apply that information. At the same time, the endurance level comes into play. And the endurance level, of course, is the issue that you need to be performing as well on that last section as far as your physical and mental stamina is concerned, as you did on the first section. So I've seen lots of students who know the stuff really, really well, who know how to attack the questions, but what they got to work on towards the end of their MCAT preparation is their endurance level. So when we think about your journey for your MCAT preparation, the earlier parts are going to be more focused on content review. The middle portion, which is really longer than an even third, I would say it's maybe 60 to 70% of your time, is going to be focused on the actual application and then building your endurance. And then the tail end is when you're just doing endurance exercises. And those endurance exercises are, of course, full-length exams. So big part of your preparation is going to be doing all of these things. Don't just jump into full-length exams right now, especially I saw from the polls, there's a large number of you that are taking the test in January. It's still not time to take full-length exams. And for those of you taking the test in March or April, it's not time to be taking full-length exams. Now is the time to be reviewing content and to be practicing the application of that content. So super, super important to kind of keep that in mind. Now, we get a lot of questions about the scoring of this exam. And the reality is the following. While it's easy to get hung up, and tell me if this is you, it's easy to get hung up in numbers, right, guys? I mean, that's not, I'm not talking out of turn when I say we get hung up in numbers. That is a normal, natural response. I see a bunch of you privately are saying, yep, that's me. So we get hung up in numbers, right? We get hung up in numbers, and we like to get hung up in numbers because numbers are easy to get hung up in. But the thing about the MCAT really comes down to this. The more questions that you get right, the higher your score will be. At the end of the day, the way to get a higher score is to get more questions correct. Plain and simple. Plain and simple. Get more questions right, and you're going to get yourself a higher score. And I know that sounds simplistic, and I've had students laugh at me where they say, oh, it's so easy for you to say that. But here's the thing. If you get hung up on the final number, you're going to only get impeded by that final number. Your mindset, and this is a little bit of a growth mindset opportunity for each and every one of you, your mindset should be, okay, what can I do next time to get that question that I got wrong? What can I do next time to get it right? What do I need to fundamentally change? Is it a content issue? Is it the answer choices? Is it my interpretation of the passages? What is it that I can be doing to make that change as far as those questions go? And you do that, you're going to be very, very happy as far as the outcomes. And it is baby steps. And sometimes with the MCAT, it's just a matter of getting 1 or 2 questions more correct in order to start jumping up on this scale. So really, I want your focus to be, especially now, kind of earlier in this process, I want your focus to be on the necessity of how to be getting more questions correct. And we're going to talk about that today when we dive into the actual session. So that's the first part. As far as the integration of the test, I alluded to this earlier. I was kind of jumping the gun, but I was alluding to this earlier. I know some of you have seen these slides already, but just to kind of, keep things in mind, I mean, look at this chemical and physical foundation section. Look at all the colors. Each color represents a different scientific topic. And in fact, you are being tested on 5 different scientific topics in that one section. There's also, and I'm just going to put this out there, there's also, and tell me if you've heard this kind of word on the street, there's this urban legend that has spread with the new exam that organic chemistry is less important than it used to be. That horrible, horrible urban legend. And I already see a bunch of you are giving me thumbs ups in the chat. It's the biggest lie, biggest myth that's out there. The MCAT used to directly test a lot of organic chemistry. Now it tests much less organic chemistry directly. However, organic chemistry is the basis, it is the alphabet, if you will, it is the vocabulary of biochemistry. And while they might not directly be testing you on as much organic chemistry as they used to, you can rest assured that all the biochemistry that they're testing you on is rooted in organic chemistry. So you're going to get a lot of OChem. So OChem still is a huge, huge part of your preparation. Now, in addition to content, you need to have certain skills that you need to be developing. And in fact, the AAMC has identified these 4 core skills as skills that they are testing on the MCAT. That is science knowledge, scientific reasoning and problem solving, experimental and research design, and data and statistical analysis. And in fact, every question on the exam, it comes at the intersection of a content area as well as one of these 4 core skills. But there's a 5th skill that's almost an implied skill that we've made explicit, and that is just MCAT expertise, knowing how to play the game, right? If you want to be good at a game or you want to be good at a sport, the better you know how to play the game and how to take advantage of the vulnerabilities of the game, the better you will do at it. And the MCAT is nothing more. I'm trivializing, but it's a game. It's a big game, and learning those rules and how to use those rules to your advantage is a huge, huge part of your success as you move forward. So we'll talk about the application of that later on here today as we go into this session. So what we're going to do, plain and simple, is we're going to do a full MCAT channel session. In fact, I taught this exact same lesson just a couple weeks ago. The MCAT channel, those sessions kind of repeat, similar to a rolling schedule, if you will. So we're gonna do the exact same session. You're gonna ask your questions as we have them through the viewing, right? And it's not even a viewing. This is live. I mean, the language on here makes it sound a little different, but it's live. You're gonna ask your questions, and you're gonna be a student. So I want you to put yourself in that mindset of you are studying for the MCAT. Now, what that also means, though, and there's a reason I'm putting my hands up here, is there are very, very distinct parameters and very distinct boundaries that you need to respect when you're studying for the MCAT. And what I mean by that is that you have learned more in school, in your undergraduate classes, about these subjects than will be tested on the exam. In fact, there are things that are, in fact, out of scope of the blueprint of the MCAT. The MCAT has a very, very defined scope, and easiest way to think about it, typical biochemistry textbook is 1,500 pages. You don't have 1,500 pages of biochemistry that you're responsible for on the MCAT. So as you start to think about this, as you're going through this experience, I want you to be really putting yourself in that mindset of, okay, I'm studying for class, and I'm looking at this in the way that I'm going to be looking at this for the actual MCAT. Okay, so that's what we're going to be doing here, and that's what we're going to accomplish. So, as I said earlier, the goal here is for you to learn a little bit about the MCAT, learn about how to prepare for the MCAT, and learn a little bit about yourself and your needs that you're going to have for this exam. But also, and I didn't say this, and I always say this, it's also to have a little bit of fun. So let's have a little bit of fun. I know it's Thursday night. What else can we do? What better thing can we be doing than having a little bit of fun with the MCAT? So let me switch us over here real quick and get our molecular genetics deck up here. Loading up. Give me a second for that to make its appearance. There we go. So everybody should be able to see the deck that says molecular genetics, DNA, and transcription. Here is what we are going to be doing. We are going to talk about the events that are involved in DNA replication. We'll talk about some of the common mechanisms of DNA repair. And then we're going to talk about the steps that are involved in transcription and in gene regulation. Okay, so these are going to be the three big things that we're going to be looking at. Now, before we go any further, I want to point something out, and that is the double T's. So write this down. Never forget this. Always be mindful of this. The double T's of molecular genetics is, what I mean by this, is the words transcription and translation. These are two of the most commonly flipped around words that you will do and you will see on the MCAT. What I mean by that is that the MCAT will actually put wrong answer choices in where they'll put the word transcription where you're thinking they should have put translation or vice versa, but then you fall for the trap. So just remember this. Transcription is taking DNA and making it into RNA. But in a more simplistic way, it's taking DNA, which is a nucleic acid, and transcribing it into another nucleic acid. So it's the same as me speaking in English and you writing everything down that I said in English. So that is a transcription. We stay within the same language. In a translation, we go from one language to another language. So translation is taking nucleic acid and moving it into amino acids. So you're literally changing scientific language. You're going from one type of acid to another type of acid. Now, it's not that you physically change the material, but you use the material, of course, to code for the next thing. So don't be fooled by that. Don't let the MCAT trip you up on that, because it will try to trip you up on that, and it's tripped up many students before in its history as far as confusing the T's. So don't, whatever you do, don't confuse the T's, because that's going to be bad, is what it's going to come down to. So let's do that. So with that said, let's take a look at what we've got here on the screen. This is our good friend DNA, and what we refer to this picture as is this is our replication fork. And the reason why we call this a replication fork is because it kind of looks like a fork. It's a two-pronged fork. Similar to what you'd stick in a piece of meat if you were, cutting it up. So when we think about DNA replication, this is neither, I want to be very, very clear here, this is neither translation nor transcription. This is replication, right? This is literally making more DNA. When we think about DNA, when are the instances that we would need to make more DNA? Not transcribing it, but actually making more of it. When does the necessity for more DNA to be made come into play? And I see a bunch of you are typing in here when you are replicating your cells, right? So that's what DNA is there for. DNA is there, and of course, when we need to have mitosis occurring, right, or meiosis, but whatever it is that we are going to make more cells, we're going to need more DNA, because the new cells, they need the DNA, right? They need the genetic information. So this is what we're looking at here as far as this replication is occurring. This is happening, of course, in the S phase, right? So the S phase is the synthesis phase as far as the cell cycle goes. You have the growth phase 1, you've got the S phase, growth phase 2, and then you got the M phase, which is the actual mitotic phase, when everything starts to separate and beautiful things start to happen. So the problem with replication is it's very, very necessary. But the fundamental problem is that you've got a lot of players that have to do their thing, and they have to do their thing very, very correctly and at the right time so that everything happens in a seamless kind of fashion. So let's go through the enzymes that are involved in DNA replication. So first and foremost, we've got helicase, and our good friend helicase does something very, very special. It is taking our double helix of the DNA, which is kind of twisted on itself, if you will, and it begins the process of pulling apart those two anti-parallel strands of DNA, right? So we're literally, it's like taking two shoestrings and starting to pull them apart, two shoestrings that are twisted on each other. The problem that we run into when we are unwinding the DNA with helicase is that we are going to start to create stress on the actual DNA in this region over here, the region directly behind where the helicase is doing its thing. And so we need to relax this, because if you think about it as two shoestrings, if you keep pulling on them, that part that's twisted gets tighter and tighter and tighter and tighter, and it's going to get harder and harder to unwind. So topoisomerase is designed to do just that. It relaxes the DNA. So you think of topoisomerase, if you want to personify it, it's kind of like the masseuse. It's relaxing the knot that's in the DNA. I know that's a horrible analogy, but hey, it might work for you. And sometimes it's just about figuring out something that that's going to work for you, and you're going to remember it. So that's that is something to keep in mind, and something to be looking at. You guys like these? You like these little corny ways of remembering things? If you don't, it's fine. I'll stop doing them, but usually people like them. So I'm gonna keep doing them until you tell me, until you're like, Petros, please stop with the corniness, because it's not working. But you'll remember this. You'll never forget this. So Ismat is saying yes. So Ismat, you're speaking for the group. We're gonna get more of these. So this is we got an hour and a half to go. So there's lots of opportunity for us. All right, let's keep going. So we've got helicase, we've got topoisomerase. DNA is nice and relaxed. It's loose. It's got the masseuse. It's done its thing. And now we have to actually start doing some work. So I jumped ahead. Sorry. Primase is going to come into play. So primase is just that. It is a primase. It is priming the pump, right? It's getting things ready to allow for the DNA replication to actually do its thing, right? So the DNA, so primase, very, very important. However, maybe the most important of all these enzymes is going to be DNA polymerase. Now, DNA polymerase, as the name suggests, is going to be the enzyme that is going to be doing the work building the new DNA. However, DNA polymerase is kind of like, think about a friend that you have or a family member who's very, very stubborn. Anybody, are you thinking of somebody who's really, really stubborn that's in your life? Anybody, maybe you have some very specific names of people that are popping in your head, and maybe your blood pressure just went up a little bit, hopefully not too much. So DNA polymerase is the stubborn enzyme, because DNA polymerase only likes to work in one direction. So when you think about DNA polymerase, think about that stubborn person that's in your life who only likes to do things in one way. Okay, a couple of you have many of these, or maybe you're the family member, right? So the DNA polymerase, what I mean by it being stubborn and only wanting to work in one direction is that DNA polymerase only knows how to build DNA in the 5' to 3' direction. Now, if you don't remember 5' and 3', that is something to review as far as content, as far as the directionality of DNA. But DNA has the two strands, and we define it as being the 5' to 3' ends, and they are anti-parallel to each other. So what's happening with DNA polymerase is that it's only going to be able to build DNA from the 5' to 3' direction, which means it is reading the template strand of the DNA in the 3' to 5' direction. But the problem that this leads to is that we end up with two different kind of flows of the creation of the DNA, of the new DNA. On the left-hand side of the screen here, you see this leading strand. And notice that the leading strand is one beautiful, continuous leading strand, right? It's one continuous strand, one kind of seamless motion of DNA polymerase doing its thing. And the reason for that is because it is building from the 5' to 3' direction, as it likes to do and as it's happy to do at any given point. However, on this other side, we've got the lagging strand, and the reason for this is because the DNA polymerase goes from 5' to 3'. But as the replication fork is opening more and more, as helicase and topoisomerase are doing their thing, it keeps jumping back to build another little piece, and jumping back to build another little piece, and jumping back to build another little piece. So it's continually jumping backwards in order to build these little tiny segments. These little segments are called, anybody for bonus points, anybody want to call out what the little tiny segments are? Give you a second or two. I'll give you 9 seconds. How about that? I'll give you 9 seconds. Type into the chat what the name is of those little tiny strands that are coming together. Those little tiny pieces. There we go. Almost got it. Almost first in. Okazaki fragments, right? Okazaki fragments. They are very good. Nicely done. There. High five. High five for you. That is not me slapping you in the face. That is me giving you a high five. So Okazaki fragments. That's what those are called. Named after Dr. Okazaki. So we've got those little pieces, and those little pieces, they got to be glued together. So we got the glue. This is the super glue, right? This is the gorilla glue of DNA. And that is, anybody want to take a guess at what the gorilla glue is of DNA? Give you another couple seconds. It is, starts with an L. Ligase, there you go, right? So ligase is going to stick all the little tiny pieces. DNA ligase, pretty important stuff. That is the gorilla glue. That's that super glue that you can buy and get everything nicely stuck together. So other things that are involved in all of this, RNA primers. Part of the reason for the RNA primer is because it's easier for the priming, the primase, and the priming to begin with the RNA primer. And then you've got some single-strand binding proteins that are there as well. Here's the thing, as far as the MCAT goes, I would focus on these 5, right? Focus on helicase, topoisomerase, primase, DNA polymerase, and DNA ligase. These 5 enzymes, these are the big ones that you should be putting some focus on as far as this test is concerned. Okay, so that is what we've got there as far as these guys are concerned. Now, the thing to remember is that this is this picture that I'm showing you, very idealized way of thinking about the replication of DNA. In fact, this is happening in multiple places. So if we were to do DNA replication like this, where it was just, one area at a time, it would take weeks and months because we've got so much genetic code. So in eukaryotes, we've got something, a little phenomenon known as multiple origins of replication. Okay, so these are really, really important because what's happening is you've got these multiple replication forks, these little bubbles that are forming throughout your DNA. And really what we have happening is these daughter strands are being formed on the inside in multiple places, and this continues on ad infinitum, and you have more and more DNA that's being built as a result of this, and faster and faster and faster. Okay, so that's what we have there as far as those multiple ORIs. Now, at the same time, as we think about our genetic code, and as we think about kind of how the genetic code is organized, of course the genetic code is built into the DNA. However, when we organize our genes and organize our DNA in time for replication to actually occur, when not the DNA replicating, but the actual cell replicating, so mitosis to occur, the DNA that's in a chromatin form is going to turn itself into chromosomes, right? It's going to organize itself, a better way of saying it, organize itself into chromosomes. And these chromosomes have these vulnerable little ends on them, right? They're at the tips. And the problem with these tips is like the bumpers on your car. This is where damage can very easily occur, right? Because it's literally at the end of these guys. So we've got telomeres. This is the adaptation that we have developed, which is a repetitive nucleotide sequence that's at the end of these chromosomes, and it's a buffer. is all it is. So telomeres are nothing more than a buffer. And so if there's damage that occurs, then really all we're going to have happen is we're going to lose nothing of relative importance. This is why you have bumpers on your car. If somebody dings your bumper, it's a relatively inexpensive repair. I'm not saying it's cheap, but relatively speaking, it's less cost than fixing the structural components of your actual car. Same thing goes with the telomeres, is that this is insurance. It is a way to protect your car or protect your DNA, your chromosomes as you have them. Okay, so if you've got questions about any of this stuff, please let us know in the chat. Are we having a little fun yet? I know we're just getting started, but are we having a little bit of fun, just a tiny bit? Or are we having like a lot of fun? Because I know we're just tip of the iceberg here. We've only hit the telomere of the fun maybe, but let's keep going with some more stuff about DNA replication. So what we know, good. A couple of you are having fun. I'm having a blast, as you can see. Shwarya is having a blast. That's what it comes. How long is this? It's about an hour. We'll be here for an hour. So you got another hour more of fun. Let's take a look at DNA replication and kind of figure out why it is what it is. DNA replication is referred to as being a semi-conservative process. Okay, it's a semi-conservative process. And the reason why it's semi-conservative is because on every newly formed double strand of DNA, you've got a parent strand and you've got a daughter strand. You have something new and something old, right? You don't have something borrowed and something blue, but you have something old, something borrowed, or something, something borrowed, something blue, something old, something new. So you've got something old, you got something new. The experiments that were done way back in the day, which you're not responsible for, but you are responsible for knowing what semi-conservative is, proved this. So they essentially tagged the DNA and they discovered that this is what we ended up with. We always had kind of half and half. There was one postulate that was out there, was that it was a fully conservative process where you would only end up with completely old and completely new. That was disproven. And then there was the dispersive, which was that it was a little bit of old, a little bit of new in any given strand of DNA. But this is the one that what actually happens. And you can see this when you look back at the replication fork, right? The replication fork gives us precisely that bit. So here's what we're going to do. We are going to test our knowledge. You guys ready to test your knowledge? See how it went. See how much we've retained. Because we've done a little bit. Let's take a look at this first question. This is it's a simple question, but a very, very typical question of the MCAT. So you might be thinking, oh, the MCAT's always going to be these convoluted questions. No, sometimes they're just like super, super simple. So here it is. The process of DNA synthesis follows this order. Now, I'm not going to have you do this problem on your own because I want to teach you how to think about this as far as test day is concerned. When you look at the answer choices, all the words in the answer choices are all words that relate to DNA synthesis's process, right? That's really what's happening. And if we think about DNA synthesis and making more DNA, what's the first thing that has to happen? Don't even look at these answer choices yet. What's the first thing that absolutely has to happen in order to get the ball rolling to make more DNA? What has to happen? Samira said it, you gotta unwind, right? Unwinding has got to happen. So here's my question to you. How many answer choices have the word unwind as the first word in the answer choices? How many of them? Just one, right? Just one. That is not coincidence, right? That is not coincidence. The coincidence would be if, well, that's not coincidence, bottom line. The MCAT, that's how they designed this test. They want you to focus on one little fact. And if you look at this and say, okay, I gotta unwind first, then it's got to be answer choice C, right? There's no other answer choice that starts with the right word. So it's easy to get caught in reading each answer choice. You're like, winding, oh, maybe that's first. Pairing, oh, maybe that's first. No, unwinding, that's it, right? Just look at the first word of each of these answer choices. you don't have to read the whole thing. You don't have to go pairing, joining, pairing, right? It's just, there's unwinding, there's pairing as far as the nucleotides are concerned, and then you join everything nice and back together again. So bottom line, it's a matter of how you can budget your time. So this is an example of a question that maybe would take you 30 seconds to answer on test day, right? That's the amount of time that you would want to budget for this kind of question. Not because it's short, right? Not because it's short, but because it just requires a small level of content and a small level of critical thinking. And I see a couple of you asking, is that a real MCAT question? Yeah, this is what they look like. I mean, there are some that are much harder than this, don't get me wrong, but there's a nice variation. So part of doing well in the MCAT is getting the points on the questions that you can like guarantee you've got those points, because you'd be surprised. If I gave you this question on your own, there'd be a good population of you that would get this question wrong, not because you didn't know it, but because you overthought it maybe, right? So it's about making sure that you have those right kind of pieces in place as you go. Let's take another look at another question. Let's look at question number 2. This is also a short one. This one we're going to do a little different. Ashwarya, are we cool with a poll? Is that gonna, we're good to go on that? I think we were talking about that earlier. I said we had a couple questions that we're going to have polls. So let's try this out. So here we go. So which is not operative word a requirement for DNA replication? Which is not a requirement for DNA replication? Let's see how it goes. See how it goes. All right. So let's take a look at this guy. It's a fun one. Deoxynucleoside triphosphates, DNTPs. When we are building that new DNA that's coming in, we've got to put in some nucleotides. But nucleotides begin as nucleosides. And in fact, they are called deoxynucleoside triphosphates. That's what they're referred to. So DNTPs, they are a requirement for DNA replication. So what can we do with answer choice A? If they are a requirement, then we can do something very, very wonderful, and that is we can eliminate answer choice A. So answer choice A is gone. The next answer choice is ATP. And I actually see that about 50% of you or so said that ATP is not a requirement for DNA. So here's my question to you. This seems like a lot of work. I mean, we had 5 enzymes that are involved in the creation of new DNA. You are literally, I mean, you got DNA polymerase, it's got to grab the deoxynucleoside triphosphate and bring it in, and you got bonds that have to be broken, and things are moving around, and Okazaki fragments have to be connected. Is that going to just happen by magic, where you just say, okay, I'm going to magically make this occur with no actual energy being used? Absolutely not. In fact, ATP is very necessary, right? Very necessary. There is no free lunch, right? There's no free lunch. Yeah, the ligase needs ATP. The polymerase needs ATP. We need energy. ATP is the energy currency, right? The energy currency as far as says, yeah, I'm seeing your answers changing because you're going, oh, wait a minute, I'm getting better at this. So we're gonna eliminate ATP. ATP is necessary for DNA replication. And then what about primers? We need primers, right? We need to kind of prime the pump as we're doing all this. So primers can be eliminated. So guess what? None of the above answer choices work. In fact, the correct answer for this one is answer choice D, because primers, ATP, and DNTPs are all necessary. They are all necessary. And I now see that 75% of you have chosen answer choice D, which is good, because that is the correct answer. But don't be fooled, right? Don't be fooled. You need all these components. So remember, there were those enzymes these are the other things that are necessary. You need ATP, you need somebody to come in and be the new nucleotide, which is going to be the nucleoside, and you need primers to make it happen. Now we have a little tradition here at Kaplan, and you're gonna participate in this tradition, and that is the tradition of receiving a very, very special thing, and that is the form of a star. So I'm gonna pick the color for this first one. I'll let you guys pick the colors for the next one. You can tell me right now what color you want for your next star, but you're gonna get a green star, right? Green star for good luck. So nicely done on the second question, your first question that you're attempting on your own. Okay, so nicely done for that one. All right, so we've done learning goal 1. Let's dive in to learning goal number 2. And learning goal number 2 is all about mechanisms of DNA repair. Now, when you take biochemistry, you're going to spend a lot of time learning the intricacies of the mechanisms and the actual mechanics of how the mechanism is believed to work, right? So these are all experiments that have been done that have led to discoveries saying this is how we think that these mechanisms of repair are gonna happen. The bottom line for the MCAT is that you need to recognize and accept that DNA does get damaged, that this is not a perfect system, and that there is a mechanism to fix the problems. So the first thing we need to remember is that the most common way that DNA gets damaged is from UV light, right? UV light, there's a reason why you should be wearing sunglasses. There's a reason why you should put suntan lotion or sunscreen, I should say. Suntan lotion almost implies that you want to get burned. So you want to put sunscreen on, right? There's a reason why these things are important, because DNA, very, very vulnerable to UV light damage. And the way that UV light or X-rays, when you go to the dentist and they take your X-ray, there's a reason they put the big lead suit on you. There's a reason why they don't take X-rays all the time. It's because it's not good for you in general. But basically what happens is that those wavelengths can cause the DNA to get damaged. And the way the damage occurs is that you break apart the bond, the hydrogen bond, that's supposed to be between A and T and C and G. And instead what ends up happening is that two nucleotides that are on the same strand of the DNA will pair with each other. And this is an example of a damaged piece of DNA. So this UV light or X-ray has led to this damaged piece of DNA. Now, instead of having a mechanism where we go and try to actually fix this piece of DNA, rather, the way the body has adapted and has evolved is we have not created a mechanism to try to fix the problem, but rather we've created a mechanism which says, let's cut out the part that's incorrect and let's try to rebuild it a second time using all the players that we talked about earlier. So good old DNA polymerase, good old DNA ligase, all the things that are necessary because we're good at replicating DNA, at making DNA. So what you have happening is you have endonuclease, or excision endonuclease, I should say, or exonuclease as it's sometimes referred to, that actually removes the damaged DNA. It literally cuts it out. This is kind of like if you got a hole in the wall, you don't necessarily just try to fix the hole, you kind of cut out a piece around it and then you put a new piece of sheetrock in or whatever. Same kind of thing, right? You're cutting out all the bad stuff and a little bit more, and then you're putting a nice clean new piece in there as you go. And so what happens is when you cut that piece out, of course, you have this very empty space. You have all these base pairs that do not have a pair with them. And so DNA polymerase will come in and it will build a new piece of DNA. There'll be this little tiny nick that's here, this little gap. And of course, ligase is going to come in and glue it together. Because what is ligase like? If topoisomerase is the masseuse, what is ligase like? Who remembers what I used as the example for ligase? It starts with a G. I'll give you guys a couple seconds to type it in. Maybe you've already forgotten. Who knows? It's super glue, right? It's the Gorilla Glue or the super glue that's going to come into place and it's going to stick all the stuff together, right? So all the stuff is going to come together. be nice and happy as a result of that. Okay, here's the good news. You might have spent weeks talking about DNA repair in your biochem classes. That's all you need to know for DNA repair. Remember I said earlier, you got to have your parameters. You got to make sure that you're focusing on the scope that the MCAT is going to actually hold you responsible for. That's the scope of it. Understanding that the repair occurs as a result of cutting things out and building anew. If you know that, you're going to ace all the questions, and probably not too many of them, but you'll ace anything that is related to DNA repair. So on that end, let's take a look at an actual question. So Shreya, let's put a poll up. Let's get everybody ready for a poll. I'll give you guys another 45 seconds or so. Put your answers in for this question. All of the following DNA replication enzymes can also be utilized in DNA excision repair, except for what? So give you guys 30 seconds. Try this out. Let's see how we do. Alrighty, so I'm seeing the results coming in, and D is winning, and it's winning by a landslide, right? It's winning by a landslide, and that's good, right? That's really good. So let's take a look at this, and let's see if you were right. So endonuclease, is that involved in DNA repair? Absolutely. In fact, we just had a whole slide about endonuclease, so we're going to keep that one. We need the ligase to stick everything back together, and we need the polymerase to make the new DNA. So really the order is endonuclease to cut things out, cut the bad stuff out, DNA polymerase to build the new piece, DNA ligase to stick it all together, make it nice and seamless. So the correct answer on this one is answer choice D. So that's the one we're going to pick. I saw some requests as far as colors from earlier. I think pink was one of the first ones that came in. So we're gonna do pink. That works for me. Let's see if I can. Well, let me rephrase that. Let me see if I can get the pink to work. Sometimes it doesn't like to agree with me on making this. So let's see if that. I think we're gonna get red. Oh, we're gonna get magenta. So here. You're gonna get a magenta star. Couldn't get the pink to work, but you got the magenta to work. So it all works, right? You get a star. You get two little stars. You get a big star and a little star. Okay, that little satellite star that's floating around on it. So with that said, nicely done. That's awesome, right? So this is how you got to be thinking about these things. Use the knowledge that you have to connect the dots as you're going through. Finally, before we go on to the third learning goal, which is the notion of transcription and gene regulation, let's talk about just this notion of the central dogma. And the central dogma is pretty simple, but also really, really important. And what we mean by this being simple is that this is how DNA that's in genes determines proteins that are going to be expressed, and the proteins that are expressed are going to give our individual characteristics, right? There's a reason why my eyes are the color brown that they are, right? There's a reason why my hair is, and what's left of it, is the color that it is, right? These are all genetic expressions that have occurred as a result of my DNA. And it's the same for you, right? We look the way we are, we have, the deficiencies and the the good things, the advantages that we have because of our genetic information. But before we go any further, I want to talk about that genetic information. So remember I was saying earlier that transcription occurs, and replication, I should say, occurs as a result of many enzymes playing, and DNA polymerase is very, very stubborn because it only likes to go in one direction. Well, we can take that analogy up a notch and look at this little thing called DNA. DNA, as awesome as DNA is, and who would agree with me that DNA is a pretty awesome thing when you think about it, right? It's got all of our code. It's what makes us us is makes us is what it is. DNA is lazy. It is maybe as awesome as it is, as epic as it is, it's lazy. And what I mean by that is it's like, you know what? I'm going to hang out in the nucleus, and if you want to deal with me, you're going to come and hang out with me here. I'm not going to come over to your house, right? It's that friend of yours. It's like, you want to see me? Come to my place, right? That's what DNA is saying, because DNA doesn't leave the nucleus. So back to our T's from before that I was saying that we oftentimes get confused. Transcription relates to taking DNA, reading it, and making RNA, mRNA specifically, but that transcription only occurs in the nucleus. If you see an answer choice on the test that implies the transcription occurred anywhere else in the cell, what can you do with that answer choice right from the get-go? And trust me on this, this will save you a lot of points. Right? This will save you a lot of points when it comes to test day, because there have been so many times where students have gotten this wrong because you just forget this one little thing, right? This one little thing. So DNA, you eliminate it, right? If there's anything, any kind of notion that DNA is moving out of the nucleus, say, no, yeah, throw it in the trash can. Exactly, throw it in the trash can. Transcription happens in the nucleus. Translation, on the other hand, happens outside of the nucleus, because the messenger RNA, it's the opposite of DNA. DNA is lazy. Messenger RNA, that's your friend that's got to run every single day, right? Messenger RNA is the one that's like, you know what? Maybe it's you that has to run every single day. So messenger RNA is like, I'm going to get my information and I'm going to run out of the cell and I'm going to do something with that information. So messenger RNA is just that. It's going out. And yes, this is for eukaryotes, obviously for eukaryotes, because prokaryotes don't have what's so important structure that we're talking about here. Eukaryotes are the ones that have got the nucleus. Prokaryotes have got no nucleus, right? So this is an important thing, of course, to keep in mind. So this is our adaptation as far as our eukaryotic cells. Now, when we look at this and we think, okay, how is this all going to come into play? Let's take a look at the actual regulation of genes. And this is important because there does come a time where you have made enough of what it is that you need to be making, right? And so you need to have some type of regulation. And these are both examples that are from prokaryotes, but the idea is the same as far as the regulation is concerned. So these are examples for you to learn the mechanism of what regulation ultimately means. So when we look at the lac operon, really what we have is we have a system in which we've got this operator region that's in play, right? This is the important region. And this RNA polymerase, it wants to get to this operator region in order to start the transcription that it needs to be doing, right? It needs to be doing to make itself, make some messenger RNA for us. However, if you've got some type of repressor on this, then we have a problem, right? We have a problem because we're not going to actually be able to express the gene that's here. Excuse me. So when we look at this, we say, okay, if you don't have an inducer, then the gene is going to be repressed because this repressor is just going to hang out right on top of this operating region. And the lactase, which is so important, what does lactase break down? Remember, lactase, the A's can be converted to ose, right? Like Petros. So you got lactase, it's breaking down lactose. So if you don't have lactase, then the lactose is just going to sit there. not going to make you feel all that good. So with the lac operon, what's essentially happening here is that if you don't have the inducer of lactose, the gene is ultimately going to be repressed. When lactose is present, though, and this is where it gets really, really cool, the lactose, which is the inducer, binds to the repressor. And so now we can see in the schematic, this repressor being bound to the inducer is now rectangular in shape. It doesn't have this little semicircle here. So it can't bind to the operator and repress anything. So now the RNA polymerase is free to come in and make a whole bunch of messenger RNA, which is then going to later be translated into lactase, and it's going to break down all of the lactose that is necessary for it to break down. So with the inducer in place, the genes, in fact, can be transcribed. So this is an example of gene regulation. At the same time, you have another type of example, and that is the trip operon. This one is a little bit more complex, So here, you've got the repressor, but the repressor can't bind on its own, right? It can't bind by itself onto the operator. So it's really kind of useless. And so without the repressor, the gene just keeps getting transcribed. So in the lac operon example over here, the gene only gets transcribed when the inducer is going to be there. And that inducer, essentially, the reason we call it the inducer is it induces the transcription. On this side here, what we have is the repressor on its own can't bind to the operator, so we just keep transcribing the gene until a co-repressor comes into play. And the co-repressor, and this is where the science is super, super cool, and where you say, oh, you know what, nature is a pretty awesome thing. The co-repressor comes into play, but the co-repressor is actually the end product. So this is an example where you're saying, okay, once I have made enough of the thing that I need to be making, it's going to now act as the co-repressor to regulate so that I don't make too much of that, because sometimes too much of a good thing is a bad thing, right? You don't want to just keep making something for no reason. So the co-repressor is the end product that partners with the repressor, and then together they form this nice little semicircle in the schematic. They can bind to that operator region, and that repressor then is going to be activated and can actually stop the transcription, right? The transcription can ultimately stop at the end of the day. This is necessary and is kind of present in a lot of places, but is necessary for homeostasis, right? Because you don't, again, if you start producing too much of certain expressed genes, that can ultimately, cause toxic buildup, and there are all sorts of things that can start going wrong, and you don't want them to go wrong as a matter of fact and as a matter of necessity as we look at these things. So as we start to go into all these pieces, we have to start thinking about transcription, and we think about transcription similar to how we thought about replication. But the fundamental thing that you need to remember is that transcription is there to make a new type of nucleic acid. So as opposed to replication, which was just to make more of DNA, now we want to be making RNA, another type of nucleic acid. So the key players here really can be identified into three different stages. So this is the initiation, elongation, and termination stages. So the components, and you might want to jot this down, or if you want to grab a screenshot. Here's my deal with screenshots: take screenshots, but make sure you use them later on. I know lots of students are like, I've got 5,000 screenshots. Well, have you reviewed them? So make sure that you review these things, or mark it in your, if you have the books, mark it in the books, or when you sign up for a class, you mark it in the books, then you know which table to go to because this material is all there. But really what's happening in transcription is you've got all these different kind of stages, but really the reason for initiation, elongation, and termination is you've got to initiate the transcription to occur. You got to get things started. Then you've got to actually do the elongation, where you're actually making the messenger RNA, and then you got to stop it, right? You got to terminate it. And this also, you will see similar types of terms when you look at translation, right? As far as the sequence of the mRNA when you're reading the triplicate code. We won't go into that today, but something that you have to kind of keep in mind. So you've got the promoter, you've got transcription factors, you have RNA polymerase, and you have ribonucleotides. The ribonucleotides, of course, are going to be the building blocks of the RNA, because you don't want deoxynucleotides, you want ribonucleotides now. In the elongation phase, you got the RNA polymerase. So similar to DNA polymerase, RNA polymerase is making more RNA. Helicase, of course, is untwisting, untying the DNA, and then more ribonucleotides, of course, are going to come into play as far as the elongation stage. And then the termination stage is just that. It's a sequence of DNA that says, hey, this is where we're going to stop actually doing the transcription. The transcription's got to stop at a certain point. Otherwise, you'd be transcribing forever. And this comes back also to the fact that DNA's got a lot of redundancies, and it kind of, has overlaps. You have genetic code that's kind of on top of each other and on top of each other along the way. So when we're looking at transcription and we're looking at the mechanism of transcription, we kind of start off in the same part of the story as we were starting off when we were looking at DNA replication. The difference here now is instead of having a replication fork, really what we have is a transcription region, right? We have a transcription fork. So we still need to open up the DNA, because if you think about the DNA as kind of being a closed book, you've got to open up the book to the pages that you actually want to be reading. You actually want to be transcribing very, very specific pages. And then at the same time, RNA, how many strands does RNA have? DNA is double-stranded. RNA is what? What is the very fundamental, important nature of good old RNA? What's the key part of this? I'll give you a couple seconds to type it into the chat. I see a bunch of you typing. It's just one, right? It's single-stranded. So if you see an answer choice that says double-stranded RNA, you might want to scratch your head and go, really? Because it's probably not going to be the right answer choice. So single-stranded RNA, good old single-stranded RNA. What's happening here, of course, is that you are going to have a coding strand of your DNA that's going to be doing the work. But really what we have to remember, sorry, go back here real quick, is that we're going to have all these regions. And you have a region that's known as the untranslated region, the UTR. If the UTR is a pretty important region, because that's actually telling us, hey, this is where we want things to begin. And then we have the untranslated region towards the end here as well. And you'll notice that we have this exon 1, exon 2, and this intron. Exons and introns are all going to be transcribed, but we're going to do a little bit of modification work to the RNA later down the line, depending on what we want to actually have for us at the end of the day. Now, similar to DNA polymerase, what direction do you think RNA polymerase is going to be doing its work? How does RNA polymerase ultimately read things and build things? Is it the same or different than DNA polymerase? What do you think? What do you think is the case there? Is it the same? Is it different? Yeah, it reads 3 to 5, builds 5 to 3. It too is pretty stubborn, right? So similar to DNA polymerase, it's a stubborn little enzyme. So I'm going to expand this a little bit. So here we go in eukaryotes. You've got the 5' cap formation. So this, again, a little bit of insurance, similar to telomeres, a little bumper. You've got the poly A tail formation, which is on the other end. So these are your two bumpers, if you will. And then you've got splicing that has to happen. And so here's what happens. You transcribe all of the stuff, but really all you care about at the end of the day are these two exons, the two parts that are going to be expressed. So what splicing does is it actually cuts out this intron, cuts out the intron, and it splices exon 1 and exon 2 together. We leave the 5' cap on, we leave the poly A tail. We've got all those pieces that are there. The reason for this, of course, is, again, protection as the messenger RNA is going to leave the nucleus, right? Because it might bump into things along the way, and we want to make sure that it's all nicely protected as it's going out. So really, really important to kind of keep in mind that when we have transcription and when we have all of these pieces kind of coming into play, really what we're looking at is we're trying to get the final product, because that final product, that finally transcribed product after it has been spliced, that is what is going to then be translated. And who does the actual translating? Who's the very important body? I shouldn't say that does the translating, but I should say it's kind of the factory, if you will, that is making the translation ultimately occur. Wants to take a guess at what that is, what that little guy is. And we're not going to talk about it today. Good old ribosomes, right? Good old ribosomes on the outside. Now, fun, easy way to remember this as far as exons and introns. Exons are extroverted. They're going to express themselves. Introns are introverted. They are not going to express themselves. However, bonus question time. Why do introns exist? Why are they there? They seem like a waste of space. Why is the intron, in fact, there? Take a guess. Type into the chat. I'm curious. Get your thoughts. Get your juices flowing. Why are those things there? They seem like a waste of space. Like, why did you waste my time? It's very simple. It's a simple, elegant solution. That's what it is. They regulate gene expression, but also, even more fun, is that those introns, remember, you got this information from a double-stranded piece of DNA. Sometimes that region of the intron on the other side of the DNA might be an exon for another gene, right? So this comes back to the fact that you've got these anti-parallel strands that are going in either direction. Sometimes those introns are a region that are important on the other side. And they are there to help with some regulation, but they're also there to help the other side, right? So maybe next time when you need to do some transcription, you're actually transcribing the other side. So these are remnants, right? Remnants of things. And then also, depending on kind of where you're starting, as far as, I mean, remember what I said earlier, our genetic code is actually kind of a multi-layered code. You have regions. If I were to look at the frame like this and then just shift the frame a little bit, I could get completely different genes that are ultimately expressed. So in a given gene, you will have a region of exons and introns, but the frames can shift. And yes, there's spaces in between, right? There's spaces in between for sure. And they do help you read, but at the same time, they are there because they might be used in another time, in another place. This is also how our DNA is, how our genetic code, I should say, overall, is in fact much more complex than the number of base pairs that we have at the end of the day. That's how we're able to cram so much information, right? So much information into this fun stuff. All right, so we've learned a lot of stuff. Now it is time for you to put this into practice, right? So put this into work. So I'm going to give you guys, I'll give you a minute on this one. It's question number 4. Take a minute. Shwari is going to put up a poll and try it out, right? Choose an answer. Let's see what we've learned. Let's put it into action, and let's end this with a bang. We're going to do 2 questions. We're going to do question 4. We'll talk about it, and then we'll do question 5. All right. So a minute. Try it out, and see you then. All right. What did I say maybe 10 minutes ago about DNA? When we were looking at that slide with the central dogma, looking at eukaryotic cells, what did I say about DNA and about its behavior and its personality, right? So we've all the different personalities today as far as our different players. What did I say about good old DNA? Key phrase that I told you, and I want you to remember this. Never forget this. It doesn't leave the nucleus because DNA is what? Starts with an L. It's lazy, right? It is your lazy friend who says, I'm not going anywhere. You look at the question, and it says, eukaryotic mRNA production occurs in the following sequence. Well, where do we make messenger RNA? Where can we transcribe the DNA to make messenger RNA? We got to do it in the nucleus. So your answer choice has to say the transcription is occurring in the nucleus. Now, I want you to be lazy because there's actually a mantra that for the last 18 plus years I have been driving into the minds of MCAT students. I'm going to drive it into your mind. And the mantra is to be efficiently lazy. Write that down in big letters in your notes and never forget that. You should be efficiently lazy when you're actually taking this test. If we look at the answer choices, what do they all have in common for the first couple of words? What do they all say? This is absolutely beautiful. And this is what we call a parallel type of structure. What does each and every single answer choice say? There's a phrase that we love. They all say, and I see a bunch of you typing it in, they all say transcription from DNA in. Well, let's look at answer choice A. Transcription from DNA in the cytoplasm. You can't get cytoplasm, right? That's bottom line. Transcription from the DNA in the nucleus. That could work. Transcription from the DNA in the ribosome. Transcription does not happen from the DNA in the ribosome. And answer choice D says transcription from DNA in the cytoplasm. The only answer choice that works, yeah, we got the patterns. We got all the patterns. But the only answer choice that works once you read past the first, 4 or 5 words, is answer choice B, because transcription cannot occur anywhere but the nucleus. So the correct answer for this one is answer choice B. I see about how many, Schweier will give me a final percentage. It was a 90% or so. My poll's a little bit slow here, so Schweier is giving me the assist on this one. But here's the deal. When I say be efficiently lazy, I also mean you don't have to read the rest of the answer choices. Once you know something is wrong, and this is an important test taking and really a life skill. Once you know something is wrong, even reading the rest of it isn't going to help you because there's no such thing as a half correct answer. So if you read the first part and you're like, oh, that part's not right, let me keep reading to see if the rest of it is all of a sudden magically correct. Even if the second half is a true statement, it can't be a correct answer because there's no such thing as an answer choice that's kind of right. It's either absolutely right or absolutely wrong. And once it's a little bit wrong, it's absolutely wrong. It's like you can't be a little bit wet. Once you get wet, whether it's a little bit or a lot, you're wet, right? That's the bottom line. So answer choice B, that's the one that you're going to want to choose. I see 95% of you chose it. We have another tradition here, and that is, of course, the stars. But we also have the black stars, which are the exclusive stars, and that is when you score super high. So I'll give you guys a black stars. That's really nicely done. So not too many of those get out there. I know Shwarya and Nikolai and Deborah and Carrie and Adriana will all attest that we don't give out a lot of black stars. There's only very few of those that get distributed in the Kaplan days, and so that's nice. Let's take a look. Let's take a look at this last question. And this last question is labeled a challenge question, and I do think you're going to find this one a little bit challenging. So it's a good one to end on and a good one to really exercise our muscle. I'll give you guys a minute. It's got a little DNA, little sequencing that you gotta do here. So give you guys a minute. Try this out. Put your answer in the poll, and then we'll look at it. Okay? All right, so this one is a little bit more difficult. So here's my first question for you. First thing that I want you to consider when looking at this question. They're giving you the base sequence of a strand of DNA, but they want you to give them the corresponding base sequence of the newly transcribed RNA. What base pair, or what nucleotide, maybe more specific in my language, what nucleotide does RNA not have? What letter should we not see if we're looking at an RNA sequence? And I see a bunch of you are typing in the letter T, right? There's no T when you're looking at RNA because RNA has uracil instead of the thymine, right? So no T for RNA. So since the answer choice, they explicitly said they wanted an answer choice that was RNA, we can't choose answer choice D because answer choice D is got a whole bunch of T's in it, right? So we can get rid of answer choice D right from the get-go, okay? So if you were tempted by answer choice D, I see about 13% of you initially were, get rid of it. It's gone. It's gone. Now, the other thing to keep in mind is that we are given the sequence of DNA in the order of 5' to 3' as is standard for it to be written. But if we want to figure out the RNA, we actually have to read backwards because remember, RNA polymerase reads 3' to 5', right? It builds 5 to 3, but it reads 3 to 5. So what you got to do is you got to say, okay, I'm going to start at C, then my first letter is going to be a what? If I have a C on my DNA, what's going to be on my RNA? I see a bunch of you are typing in a G. You're absolutely right. So the first letter's got to be a G, so I can get rid of answer choice C right from the get-go. I got rid of 2 answer choices without doing any work really, right? Just knowing my rules as far as RNA's nucleotides and knowing that I have to start over here. If I had started over on this side, I would think, it's C, right? Because I got a G, so I got to put a C in. Therefore, the correct answer must be C because that's the only one that has C as the first letter. But remember, you got to read it 3 to 5. You got to go backwards as far as the reading is concerned. So we know that it's going to be G. The T is going to give us an A, which doesn't help us because both answer choices A and B both have an A as the second letter. So that one doesn't help us. We go to the third nucleotide, which is a G, and that G is going to give us another C. So the correct answer is not going to be answer choice A, it's going to be answer choice B, right? Answer choice B is going to be the one that we're going to want to choose. And that's the one that you're going to choose. That's the one that's going to get right on test day. You don't need to read the rest of it, right? Beyond that, it's useless for you to read it once you're able to eliminate. So another kind of key takeaway is sometimes elimination actually is your best friend, especially on a question like this, which has got this long genetic sequence that comes into place, and that's an important thing to keep in mind from that perspective. So important to remember the rules as far as RNA polymerase. Important to know those rules as we're going. Adim, you asked the question of how do we know if it isn't an RNA-like strand? I'm not sure what you mean by that question. So maybe you can clarify that for us. The question said to us that we start with DNA and we are transcribing it to RNA, which is why we are able to apply the rules that I just applied as far as the rules as far as uracil needing to be present since we're making RNA, or rather thymine not being present. It's very possible that that uracil isn't present. That doesn't mean you don't have RNA. That just means you have a piece of sequence that doesn't have uracil in it, right? So you just know that thymine can't be present when you're looking at an RNA type sequence. So really important to keep that in mind. Now that's the lesson. So you have now literally gotten the exact same lesson that we deliver on the MCAT channel related to molecular genetics and transcription. There's obviously other things to talk about, but in this lesson, as far as the focus for this lesson, that is what we cover. So what do you guys think as far as molecular genetics and the MCAT and transcription? There's obviously translation to come, but as far as what we saw here, would you say it's manageable? What do you think? Is it manageable? Is it doable? going to be okay, hopefully. Have you had a little bit of fun? is maybe the question that I'm asking. So good. So yeah, so this is, I mean, it's totally doable. The thing that makes it intimidating, and I share this sentiment with you, what makes it intimidating is there are lots and lots of players that come into play, right? There's so many variants and things. And of course, we did this together, right? We walked through this. Now it's a matter of being able to execute this on the test and execute it within the time frame that you are given and under the stress that you are going to be under. Because that's a big part of it for me too, is that, yeah, you might know a lot of stuff, but at the same time, sometimes that stress comes into play, right? And all of a sudden you're like, oh no, what am I going to do? I only got 5 minutes left, and now, I got to execute on this stuff. So at the same time, there is this issue, and we're going to spend a little time kind of talking about some fun stuff now, not really content related per se, but just how do we deal with the after effect? So a big part of your doing well on this exam is going to be doing lots of questions, right? Doing questions, maybe the best thing that you can do for yourself, because it's by doing practice problems that really gives you that opportunity to put into practice the things that you've learned. I've had students in the past who've known the content better than anybody that I know. That includes teachers, includes my professors. I'm just like, you are a content master. You can recite the textbook. But they've struggled not because they didn't know the material, but they didn't know how to apply the material, right? Imagine having this wealth of information, but not ever actually being able to put it to good use, right? To put it into application. So big part of your practice is going to be to actually put what you know into action. And that's really, really a key part of this. So definitely get it into your plan early on when you're learning content and you're reviewing content. You want to be reviewing the content and then backing it up with a couple of questions that reinforce your knowledge. It's not questions just so that you take a question, it's so that you can retain that information longer at the end of the day. And so the big question that I get all the time is, how can I actually retain information? The way you retain information is by practicing it, by applying that information, by being able to put it into the right context, if you will. At the same time, it's super important to make sure that you're not over practicing with questions without doing something different before you do more questions. So a big part of being successful on this exam comes down to being able to identify your patterns and your areas of opportunity. And we do it in a couple of different ways. So in our course, you have a series of recommendations that are given to you, that literally, we have a really awesome algorithm that's weirdly getting smarter. The more people that use it, the smarter it's getting, where it actually will say, okay, based on your performance, based on the questions that you got right, based on the questions you got wrong, here is what we are recommending for you to be doing next. So literally pointing you to specific assignments, specific material, specific exercises of what you should be doing next in order to improve your score. So we call it smart reports, but it's really just this adaptive learning engine that's doing this awesome thing of gathering all this information and really being able to kind of tell you, okay, here's what you need to do next. But part of it also is a manual process. And the reason it being a manual process is because by you actually taking the action yourself, the studies have shown that you have a greater chance of improvement by being able to start to extrapolate information about what your individual needs are. So we recommend this really simple, but very, very awesome system, which I'm going to walk you through here, which what we call it the Why I Missed It sheet, the WEMIS, as it's known around the office. So, I was talking to Adriana the other day. I was like, well, let's go through your WEMIS, right? And that's how we talk, is we talk in acronyms, talk about the MCAT, talk about the WEMIS. The WEMIS is the Why I Missed It sheet. And bottom line, what this is an ongoing record of the questions that you have gotten wrong. So at the beginning, you take the diagnostic exam, right? Or maybe you took a practice test with us this past week. The best thing that you can do for yourself as a first step is to go through the test, and you should review the entire test. You should review the questions that you got right, the questions that you got wrong, but you want to maybe spend a little more time on the questions that you got wrong. Okay, so that's kind of the first area to kind of get you into the mood and get you into the flow of studying. But as you're going through any set of questions, whether it's a full-length exam or a small set of questions, or if it's in the adaptive Q bank, whatever it may be, what you want to be doing is you want to be documenting where the questions came from. So, in this case here, it came from the diagnostic exam, it came from this section, this was the question number. You want to document the type of question it was, if it was a discrete standalone question, or was it a passage-based question, the content area that it was testing you on, or the type of question when you're looking at the cars section specifically. And then you want to make a personal note of why you missed it. So did you read the answer incorrectly? Did you not know the content? Whatever that reason was, you want to jot that down. And you can literally do this in an Excel file. Like there's no Kaplan form, there's no Kaplan why I missed it sheet that comes out. It's an Excel file. So just make 6 columns, exam section, question number, passage based or not, content type, why I missed it, and you're done. It can be a Google Doc, whatever, spreadsheet form that you like to use it. And then what you do is you kind of build this out. And so as you're post-phrasing, you're summarizing why you missed it. And as this list gets longer and longer, you can start to identify patterns. So let's do a little exercise here. So this is a real student. So real questions that they got right throughout their Chem Phys section of the diagnostic. What would you say is an area, just based on this information? I mean, we've got some content areas here. We've got the reasons why they got the questions wrong. Take a minute, analyze this, and see why is it. So Simone's starting one right now. I love it, right? Start one ASAP. Start looking at this student and tell me what patterns or what things you would recommend for this student to work on ASAP. Like what stands out at you on this little example that we have here on the screen. Again, this is not meant to be more complicated than it is. This is relatively straightforward. Marla, the SMART reporting system, that is part of the course. So when you're enrolled in our program, that is built in. It's a really awesome system. So really, really awesome. Super, super smart. Kind of scary smart is what it is. So I see a couple of you are typing in carbs and misread the correct answer or didn't know the content. So yeah, let's look at this. I mean, here's a student, question 18, 19, and 20. Probably they were all passage based, right? They were all they were linked to a passage. Passage was probably about carbohydrates. In fact, I know that in the diagnostic, those questions are about carbohydrates. So they didn't get any of them right. And for 2 of them, they didn't know the content. And then for the 3rd one, they misread the passage detail that was necessary to get the question right. So on a grand scale, first thing this student should probably be hitting the books on is carbohydrates. Right? Carbohydrates is bottom line, plain and simple. Now, this is the diagnostic exam that's from our course. That's the same diagnostic that's in that free trial that I told you about earlier, right? So if you want to take the diagnostic that's in the course, sign up for the free trial, you can take the diagnostic. You might have taken the practice test. That's a different test. Practice test is not the diagnostic that's in the course. So if you want to, see how you do on question 18, 19, and 20 about carbohydrates, you can try it yourself is what you can do. At the same time, there's another pattern here. And I see a bunch of you said this as well, and that's just not knowing the content. So knowing the content for these areas indicates to me, if I was coaching the student, working with the student, I would say, okay, you didn't get those things right. You got to go relearn that content. You got to review that content. And there are lots and lots of resources, right? Lots and lots of resources that you could, that you can be going to within the course, within the overall program, I should say, that will help you build up on that content. Maybe you need to reread something, but more importantly, maybe you need to see a video of it, right? Maybe you need to see a little short video demonstrating whatever VESPER area it is that you have missed. So the idea behind the why I missed it sheet is that the sheet will get longer because at the end of the day, you're going to have every single question that you've gotten wrong. And we've got over 10,000 questions in our course. So you're going to have, a good number of questions that everybody gets questions wrong. You're going to have questions that you got wrong on your why I missed it sheet. And you can use that to start identifying what your patterns are. And that can help you start to focus on what it is that you need to be working on as far as improving your score, right? So very, very important to keep that in mind, and very, very important to kind of think about this in the longer term. Now, what you will see the more you work with the why I missed it sheets is the reasons why you're getting things wrong will start to, gravitate towards a couple of bigger areas that then you'll be able to start to completely eliminate from your issue. So what might happen is you might say, you know what? I've now focused so much on carbohydrates that I'm getting every single carbohydrate question correct. And this comes back to the second area that I wanted to focus on for our kind of final minutes here together, is that it's easy to study the things. Who would agree with this statement? It's easy and rewarding to study the things that we're really, really good at. Like if you're getting every single carbohydrate question right, you're like, I'm going to study more carbohydrates. And you keep doing carbohydrate questions and you keep getting them right. And you feel really good about yourself. You're like, I'm a rock star. I'm awesome on the MCAT. I'm getting every single carbohydrate question correct. Who feels that sometimes? Or maybe this has happened to you recently, right? And this is true in life as well, right? It's true in life as well. We gravitate towards hobbies that we are good at, right? We gravitate towards sports that we are good at. And we ignore or we don't do the things that we're not so great at. The problem, it's a positive feedback loop, right? You're like, I feel really good about myself. I'm going to do some more carbohydrates because I'm going to feel really good about myself. And you just keep doing more and more carbohydrates. The problem that you run into on the MCAT is that there are only so many questions that are going to test you about carbohydrates. So if you're acing every single question about carbohydrates, it's time to move on, right? It's time to move on to something else, right? It's time to move on to the next subject area. So as much as we don't like looking at things that we are not good at or that we struggle with, the place that you can improve your MCAT score, is not necessarily in the areas that you're already good at. It's in the areas that you're not so great at. And this is the hardest part of studying for the MCAT. You have to force yourself to work on stuff that you don't like working on. And that's just reality. If you want to, there are only so many points that you can get from a given subject area. You got to force yourself into areas that you're just not as comfortable with. The problem that we run into when we're studying unguided is that we tend to go back and just keep doing stuff that we're good at, and we feel good about ourselves and we feel like we're doing something, and then we go take a full-length exam or we go take the real thing and we don't perform as well as we thought we would because we didn't spend the right time studying the right places. So my advice to you, and this comes from the whole team, so this isn't just me, this is all of us and everybody at Kaplan, all the faculty, we say this to our students all the time. It's about making sure that you are doing the work that is right for you. The personalization level that is necessary for the MCAT is absolutely key. And one of the biggest things that bugs me, and I say this in a positive way, I'm saying it with a smile on my face, it bugs me, is when I hear students say, well, my friend studied X, Y, and Z, and she did really well on the MCAT, so I'm going to do the same thing. And I usually will turn to the student and say, that's great for your friend, but isn't necessarily right for you. So that, it's not about just doing what other people did. It's about doing what is right for you. This is your time that you're investing. You need to be making sure that you're doing the work that is absolutely right for your individual situation. We personalize the course based on your needs. So those recommendations I was telling you about in the SMART reporting system does exactly that. It actually says, here's where you, Ashwarya, need to work on what you need to work on. Here's Nikolai, Deborah, Adriana, here's what you need to work on. Carrie, this is what you need to work on in order to improve your individual score. It's also not just about the number of hours that you're spending. You want to make sure that when you're building your study schedule, you're building the study schedule to be specific to work that you individually need to do. It needs to be realistic as far as how much work you're going to do in a given amount of time, but it also needs to be adaptable, because there's going to come a point where you're going to be getting every single carbohydrate question correct, and then it's time to move on to the next thing. And it's going to be a point where you say, you know what, I've done enough reading, I've done enough reviewing, I got to start doing some questions, or I got to start taking some section tests, or I got to start taking some full-length exams. And it's really about making sure that you structure that progression towards test day, so that you're not all of a sudden, first time looking at a full-length exam is the actual MCAT. It's very disheartening to me when I hear from students who are like, oh, my mistake was I didn't take a full-length exam before I took the actual exam, which is sometimes shocking to hear that. But then it's like, all right, well, come take our course, and we're gonna fix that along the way. The other thing about the MCAT is that you don't want to take this test too many times. So really, really easy to fall in the trap of, I'll just repeat it. Med schools see all the scores, right? They might only consider the highest score, but it's sitting there. It's a couple of friends who are admissions officers, like they're like, yes, we only consider the highest score, but we see the other scores. Like, it's in the corner of our eye as far as the decisions are concerned. So really, really important to make sure that you do that in the right way as it goes. Okay, so something to kind of keep in mind. So look for trends, look for patterns in your mistakes, and you're gonna be nice and happy is what it comes down to. All this information is on our website if you want to find out a little bit more about what's offered in our courses. The slide is actually technically out of date. We now have 15 full-length exams because the AMC has released another full-length practice test. So that is now included as part of our courses. But bottom line, we've got a whole big program, and there are many, many more hours of material than you are going to individually need. But we've given you all the stuffs so that you can, again, using the SMART reporting system, using kind of where your individual strengths and weaknesses are, you can go to the lessons and go to the sessions and use the materials that you ultimately need to do. What you saw today was the MCAT channel. So, the MCAT channel is just this. They're hour-long sessions. We've got a ton of them. Those are in addition to your core sessions that you have as part of the course. Is how we actually have the most live instruction that's available to any student that's out there preparing for the MCAT. So really, really awesome to kind of keep that in mind as we go. But here's what I thought we would do for our last 15 minutes or so together. We've got a veteran team of teachers that are in this room. So, and I'm lucky because I get to work with Adriana and Ashwarya and Deborah and Carrie and Nikolai all the time. And you're really lucky because they're all here and I'm here, and I'm not, sorry, that came out wrong. What I mean is, I want you to pick our brains about how to study for the MCAT and how to study for this all important exam. So what I'd love to do is just make this next 15 minutes or so kind of an ask us anything. Ask us what's on your mind as far as building out the right kind of structure, the right kind of plan as far as your individual needs are. What's burdening you on your mind? I mean, some of you have been coming to all these sessions this week. I'd love to answer whatever questions, and I know Adriana, Deborah, Nikolai, Ashwarya, Carrie, we'd all love to answer these questions as we go. So start throwing them in. Like, just let us know right now what's on your mind. What is it that you're, what is it that you're worried about? What is it that what you're taking? Adrian, I see you definitely don't want to pay $315 a second time to take the MCAT. Not a good thing, right? You don't want to do that for sure as well. It's an expensive practice test is what it comes down to if you're like, oh, I'm just going to take a couple times. Peyton, great question. Can I compare in-person versus live online classes? I sure can. So to be perfectly transparent, I started as an in-person instructor at Kaplan because we didn't have live online classes. And then maybe now 9 years ago or so, we launched our live online programs. And I can say, having taught in both environments, the live online experience, oddly enough, is actually more interactive than an in-person experience. And the reason for that, similar to what you see here today, is you've got more than one instructor that's in the room. So if you've got a question, you just ask your question, right? You don't have to raise your hand, wait to be called on, et cetera, et cetera. The materials that you receive are the same. So regardless of, what you're getting, you're always getting the same materials, regardless of the type of course it is that you're taking. But what the live online experience allows you to do is allows you to have that greater level of interaction, and it gives you the opportunity to actually interact with a population of instructors that you might not have been able to interact with, because for our live online classes, we actually draw from the entire country. In fact, technically internationally, because we have some teachers who are from Canada. But we're drawing from our faculty pool that's wider spread. And so you're actually able to get that exposure. Now, everybody gets the MCAT channel. So, and the MCAT channel is live online only. So even if you're an in-person class, you're going to have the MCAT channel experience for sure. So, that's, I think, the big, big difference between the two is that level of interactivity and the convenience, quite frankly. You don't have to go anywhere. You don't have to drive to a specific location. Selfishly speaking, as a student, I like to get my questions answered, and I don't necessarily want to wait for somebody else's question to get answered. And that's a big advantage with live online, is that you do have that more immediate attention. So as I said, it's a little more interactive than such. Parshvi, hopefully I pronounced your name correctly. The 15 full-length exams that we have, 4 of them are from the AMC. So 4 of them are from the actual test makers. The AMC has released 4 full-length exams. The other 11 are written by Kaplan. So they are our creation. However, over the last 3 years, as the MCAT has been more and more administered, right? So the more administrations we've had of the actual MCAT, we've actually taken the 11 full-length exams and done a full, and I'm going to use a very, very fancy term here, a full psychometric review, which is a very, very fancy way of saying we have analyzed how accurate our full lengths are to the real full length, to the real exam, and made them more and more real. And we have now, actually, for more than a couple of years now, we've gotten to a point where the confidence level of our full length exams compared to the real thing is so close that they're indistinguishable from each other. So they are phenomenal resources for practice. I hear rumors all the time. I read the same forums that you guys read. Kaplan tests are harder. Kaplan tests are easier. Kaplan tests are, they look different, or the AMC looks different, whatever it might be. We hear the equal amounts on both sides. The reality is that when you put them side by side, they are indistinguishable from each other. We build our full-length exams off of the blueprint of the actual AMC. And by blueprint, I mean publicly available. So it's not like we have any type of, inside handshake agreement. Publicly available information that lays out what you are responsible for, right? What you are responsible for on the exam. And that's how we build our exam. The AMC releases this every year. They update it every year. We not only update the exams, we update all of our materials, so including the full lengths, all 10,000 questions. In fact, just this past summer, when they released the blueprint for the 2018 exam, we threw out questions that no longer were relevant for the 2018 exam. And at the same time, we brought in some new questions that would be relevant. We took stuff out of the review books that were no longer going to be tested. So bottom line, it's one of those things we take a lot of pride, and actually one of my key focuses as the director of pre-health programs is that we want our program to be as realistic as we can be for our students. In fact, one of the fun things we did, so this is a total aside, but I think you guys will appreciate this. We actually went into the AMC's practice tests, and we even tried to match the color of the font and the font is black, but there are different shades of black, right? There are hundreds of different shades of black that you can have. We even got the font down, right? I mean, with, just like I personally like living in a culture of, or in a world of no surprises, and I want my students to do the same as well. And we want all of our students to do the same. So that's really important to kind of keep in mind as well. So something to keep in mind, and something to 50 Shades of Black. There you go. I was about to say it, and then I was like, I shouldn't say it, but there it happened. So that's so there's that part of it that definitely is super important. The question from Shahad. Do the in-person people get the MCAT channel where they can view these live lectures? And the answer is yes. So when you are enrolled in an in-person class or a live online class, whether it's in-person or in-person plus, or live online plus, or if you're enrolled in self-paced plus, you get access to the MCAT channel. And the MCAT channel are those live sessions that are happening. Literally, there's a schedule, and when you sign up for the trial, you'll even get a sampling of the MCAT channel schedule. So you can see kind of, we have a PDF that shows you kind of what your typical week looks like. And the nice thing about it is that it kind of rotates. So if you miss molecular genetics part 1, you can come to it the next week. And we have them all recorded as well. So you have, you can always, you can always get them on a recorded fashion if you you don't want to wait or whatever it might be. But yeah, it's the bottom line. It's available to all of our students as part of the course. The in-person classes are just one teacher. Yeah, it's just one teacher in the room. It'd be a little awkward to have multiple teachers in the front of the room, but we can try that maybe. But I think there might be some more problems that will create than solutions that it will create. Definitely in the long run. So something to keep in mind as well. That's an important part to keep in mind. I'll put this slide up. I didn't want to go through the slides just for the sake of going through the slides, but you were asking. So, bottom line, we'll help you figure out the right instruction that's for you. So I don't want to necessarily say that live online is the right instruction. I'm not going to say that in-person is the right instruction. I'm not going to say that self-paced is the right instruction. I'm not going to say that private tutoring is the right instruction for you. Everyone's a little different, right? So remember what I said earlier, what worked for your friend might not work for you. So you want to make sure that you're picking the right thing. So easy way to do that, you can email us at consultateacher@kaplan.com. You can call us 1-800-CAP-TEST and actually talk to one of our faculty members who can kind of help you figure out what's right for you, right? It's and the nice thing is, and I want everyone to be aware of this, and we have this happen all the time. Student chooses an in-person class and goes, not for me, I want live online. Or student picks a live online class and goes, I actually kind of want to go to a class in person because that helps me. So, bottom line, we'll help you find the thing that's right for you. Don't feel like you've got to do it. So at that point, Shahad, it's the same lessons aren't repeated weekly per se. There are currently about 100 or so MCAT channel sessions. So they rotate. So, we think of it as going from 1 to 100, and depending on the time of year and depending on the frequency that we're running the sessions, usually the rotation is about once every 2 or 3 weeks is when a session will, come up again. But it'll be offered many times within your period of study. So you'll never, you'll always, it just kind of goes on a rotating, ongoing basis. So that's the key there. The self-paced course does not have the MCAT channel. Self-paced plus has MCAT channel. In fact, this was the slide I was trying to get to. There it is. This kind of shows you what has what, if you were curious to know, do I get this? Do I get that? So self-paced plus has MCAT channel, but self-paced does not have MCAT channel. So that is one of the big differences between that. The plus is where you get 3 hours of individualized one-on-one coaching with one of our MCAT experts. Our MCAT experts, just to give you a little bit about their profile, they are veteran MCAT faculty who have been accepted to medical school. So these are folks that are, have been with us for some time and have, taken that next step as far as the process goes. The coaching is about coaching you the right way through the process. It's not private tutoring. Private tutoring is, like when I tutor a student, that means they come and they say, okay, let's go over force equals mass times acceleration. We go over force equals mass times acceleration. When I coach a student, it's, all right, are you doing the right things at the right time for you? It's this conversation, but in a more personalized one-on-one way. So one of the things that we talk about in coaching is building the why I missed a cheat, which is why you got the sample there today. And you've gotten other samples previously in the week of kind of the things that happen as far as the coaching time is concerned. Other questions that you guys have? See what else you have in here, and maybe if there are any that have come in that I can answer for you. Final thoughts. Megan, yes, Megan, you do get the 7-subject book kit when you enroll in the class, so you don't need to go and buy that on your own. That is included. In fact, you get 10 books. You get the lesson book, you get the quick sheets, and then you get this really awesome book, which is called the High Yield Problem Solving Guide. We developed the first version of this. It's now on like edition 10 or whatever. But the first version of it, when we were creating it, our code word, because we didn't want anybody at Kaplan to know what we were working on. It was kind of a little bit of a side project that we wanted to, eventually launch out en masse. This was over 10 years ago. We called it the Kaplan MCAT Cookbook. Everybody thought that the MCAT team was collecting favored recipes from students and from faculty, where in fact what we were collecting were the most common questions that show up on the MCAT and the way to solve those. And the example I love to use is KSP problems. So if you're solving for your solubility product, there's really one way to solve a KSP problem, right, or a KEQ problem, regardless of what elements or what molecules are involved and what the concentrations are. So we literally built out this awesome book, which step by step shows you how to solve the highest yield questions that are in the course. So you get that book as well. So, and then the 7-subject books, you get a big box. It's a very heavy box, but you also get them in a digital edition as well. So you have the kind of option to use them as you like as far as your coursework is concerned. So hopefully that answers your question accordingly. And if not, let us know. Any other thoughts before we wrap up here? Because it is 11 o'clock. It is late. I don't want to keep anybody. You're welcome. You're welcome, Megan. And good to see you today. I know you and I were emailing a couple days ago with some questions. So good to see you here again today. So with that said, I wanted to take this opportunity to thank the team. They've been answering your questions. I know a lot of you were asking private questions, so they've been answering your questions. So Adriana, Shwarya, Deborah, Carrie, and Nikolai, from me to you, big thank you for being here this Thursday evening from both myself, and I'm sure I speak for everybody here. A big thank you for your awesomeness. And to each and every one of you, I wanted to thank you for joining us. But more importantly, just to kind of close this out, at the beginning I said, you're going to learn a little bit about the MCAT. You're going to learn a little bit about kind of how to study and structure the MCAT, as well as we're going to go through some content. And hopefully you'll learn something a little bit about yourself and about kind of what your needs are going to be and how to structure your time and have a little fun doing it. So I hope we've accomplished all 4 of those goals. I think we have. I'm a little biased, but I think we have. I sense that we have from the chat. You're all very, very welcome indeed, and that welcome comes from the whole team as well. There are also a number of folks who are behind the scenes that I just wanted to thank as well, and that's Greg and Nick, who, to make something like this happen requires a small little village, and so Greg and Nick are that little village that makes this happen. So big thank you, and a number of other folks that get the tech to work as well. So big thank you to all of them on your behalf as well. With that said, remember, sign up for the free trial. Try it out. Remember the code is MCAT250. So if you're ready to sign up, now's the time to do so. And then at the same time, you can always book some time using that Kaplan Teacher Consult .youcanbookme link, or you can always email us at consultateacher@kaplan.com. So with that said, have a great rest of your Thursday. I hope our paths cross again. Best of luck on the MCAT, and I wish you all the greatest success as you go towards that path of wearing a white coat. Thanks, guys. Have a great evening.